senses. So sleep, of course, is required for survival, even in the long term, in a sense that you can learn some strategies that help you survive better, because you're going to be smarter and more skilled. But we mean literally in the short term. Why is it that you need to sleep? Because if you're prevented from sleeping, you're going to die. This has been shown in many model organisms. So there is this. There is a sense that sleep is essential for survival, but there's no answer for why. And this is something that we wanted to answer. And the work that I'm going to present here is of two really amazing postdocs in my lab, Alex Vaccaro and Yossi Kaplan-Dor. And they were also really, they were helped a lot by a really, really talented technician in my lab, Keishi Nambara, and a bunch of undergrads that I'll acknowledge at the end. So what Alex and Yossi wanted to do was to answer this question, and the reason why we thought that there would be this is a good opportunity to sort of take a new approach is because people have studied question of sleep and survival for a really long time, and for the most part, people focus on the brain and the nervous system when they think about sleep, and there's good reasons for this. So when you sleep, first of all, you need the nervous system to fall asleep, and when you sleep, you dream. And if you're prevented from sleeping, you really have many brain-related problems even the next day. You can't really think straight, you can't remember, etc. And it's been shown that sleep is indeed required for many processes that are occurring in the nervous system, like synaptic homeostasis, clearance of various molecules from the brain, even dynamics of chromosomes in neurons, clearance of damaged axons, etc. But then there's this whole other side that is often ignored when it comes to sleep research, which is that if you're prevented from sleeping, there are many other kinds of problems that occur in the body and that don't seemingly have anything to do with the brain. And so metabolism is altered, there's endocrine disruption, cardiovascular problems, immune system problems, etc. So this to us suggested that sleep probably is, excuse me, is playing roles that are not just centered on the nervous. And most directly, I would say people have tried for many decades. People have tried to figure out why sleep-deprived animals decline and eventually even die if they're prevented from sleeping for long enough. And they could never find anything in the nervous system that could explain this phenomenon. So we just thought to take a more unbiased approach and to say, well, let's first see if animals are really dying when they go to when they're prevented from sleeping, and if this is the case, then let's think about the possibility that this could be coming from anywhere in the body. Again, considering the fact that lack of sleep leads to many body-wide problems, and the fact that you can't really find anything in the brain of sleep-deprived animals that could tell you why they're dying, and the fact that animals that are much simpler than us, such as these jellyfish, and then, of course, all the way to mammals, mice and us, sleep. So these animals don't have a brain. They don't have the brain. They don't have the same brain that we do. This is, we didn't just appear here with this big, beautiful brains. We had to, go through this whole process of evolution. And these animals, the very early animals, they actually sleep. They show this behavior that is similar to sleep. And I think, sometimes people say, well, if you look at their electrical activity, it wouldn't be the same as ours. And I really don't care about that. The fact is that these animals engage in a behavior where they disconnect for many hours at a time. They ignore their environment. They're definitely in a different state. And I think that that tells us that sleep, originated early on and then probably acquired many more functions as animals became more complex. But there should be some primary or primal reason for sleep that is still there. All right, so this is a fun quote from this guy, Alan Rechtschaffen, who was actually one of the first people to do sleep deprivation studies in rats. And he showed that sleep-deprived rats die prematurely, but they couldn't figure out why they were dying. It just seemed like there were body-wide problems, but they couldn't figure out what was going on. And he said that if sleep does not serve an absolute vital function, then it is the biggest mistake the evolutionary process ever made. Because, of course, you spend many hours sleeping, so you're seemingly unproductive and vulnerable during this time, etc. So there has to be a reason for it, and we're after that reason. So, okay, so this is what we do in the lab. We started off with flies, and we use flies because, of course, they're the best genetic model organisms. You can do all kinds of tricks in flies that you can do in other organisms, and most importantly, their sleep and circadian rhythms are very similar to ours. And this is how we study actually sleep. We put flies in these cuvettes where they have a lot of room to walk around. This is food, and then there's these multiple infrared, light beams which go across the tube, which the flies cannot see. But when they walk back and forth, they break these beams, and that's recorded as movement. So here we see when flies are in cycles of light and darkness. This is 1 day of data collection. Okay, then so this is day 1, day 2, etc. This is the first part of the day, the light part of the day, the dark part of the day, and you can see that flies are more active during the light part and less active during the dark part of the day. So they have consolidated rest and activity periods. You could, of course, say that when an animal is in this period and is not moving, it doesn't mean that it's actually sleeping. It could just be not moving. But people have shown that flies that have been quiescent for 5 minutes or more have an elevated arousal threshold, meaning that they don't respond to the same stimuli that they do when they're sleeping, the same way, as when they're awake. So the same thing that happens to us as when we are sleeping. And I'll come back to this part later, hopefully. So flight sleep is under circadian regulation. They are primarily sleeping during the night and more active during the day. Although, as I'll show you, male flies have a big period of sleep during the day as well. And then fly sleep is under homeostatic regulation as well, meaning that if we sleep deprive them during the night. So here, during this night, flies are on a shaker. And here, actually, this is a little bit too much stimulation, I would say, but here they're shaken and they're forced to walk around the whole night. And then the next day they are sleeping during the time that they would normally be active. Here. Their brain electrical activity changes that occur with fly sleep. As I said, this is one area where it's difficult to compare flies and mammals. The exact signature, electrical signatures in the brain are not going to be the same, but there are changes in the fly brain, so they enter this state of disconnect. And then the conserved molecular pathways guide sleep in both flies and mammals. Not that we know too much about it, but the things that we know, it seems that the same pathways are involved, and then the same things occur with aging in flies, and in mammals, sleep becomes more fragmented, less abundant, etc. So they're an established model system in which we study sleep. And so this is how we prevent flies from sleeping. We actually use 3 different methods, but I'm going to focus on one here and just mention the other 2. So what we do is we try to target small populations of neurons in the brain. So here, or in the nervous system, here is the brain and the ventral nerve cord, and in green are neurons that are labeled by this particular Galford lines, and you can see the Galford line, and you can see that there's not that many cells that are labeled. And so what we want to do is activate these cells, and these particular cells we want to activate because they've been shown by Bill Joyner's group to prevent sleep. These are neurons that if you activate them, you can actually suppress sleep. And we have multiple such lines that we can use where we can target presumably non-overlapping groups of neurons, activate them, suppress sleep, and then ask what happens to animals. And we do that with a thermogenetic method where we put heat-sensitive cation channel TRPA1 inside these neurons. And then when we switch temperature, so it's like optogenetics, but instead of using light, we use temperature. When we switch temperature to 28 Celsius or above, we actually activate these neurons, these green neurons here that are expressed in TRPA1. And this is what it looks like. So we're looking here at sleep, again, across one single day. This is the light portion, dark portion. In grays are going to be our parental controls, and then in the green are experimental animals. And same here in blue. So here are two different conditions where we're targeting two separate populations of neurons, but we're losing or we're suppressing the same amount of sleep. And then we want to compare these two conditions. And we want to see if we see the same thing in sleep-deprived animals that are deprived by activating group A of neurons or group B of neurons here. And so before we switch the temperature, we see, like I said, the male flies have this big sleep out during the day, or what's called siesta, and they also sleep during the night. And for technical reasons, we're using males here. I'm not going to get into that. But what you see is that, again, this is inverse of activity. You see that flies are active early in the day, then they sleep in the middle of the day, then they're active again at the transition from light to dark, and then they sleep again during the night. If we switch the temperature to 29 degrees Celsius, you can see that the controls in gray here, they continue sleeping. There's a little bit of suppression of sleep during the night. So this is lower here than it's here at 29 degrees. So heat itself will suppress some of the sleep. But you see that that's nothing like what you see in the experimental animals, this green here, where sleep is essentially at the floor. When you activate a particular group of neurons. When you activate this group of neurons, you also see the same thing. So in both of these cases, we're losing about 90% of sleep, or flies are losing about 90% of sleep. And if we compare those two conditions in terms of survival, we see again that they behave similarly, where the control animals living up to 40 days at this temperature, the experimental animals that are sleep deprived are all dead by day 20. And so here we're plotting population survival, and here you can see survival or day of death actually plotted for individual animals. And what you can see here for the green and blue genotypes, and these are the names of L4 lines that we're using. You see that around day 10, there's this massive uptick in death. And like I said, by day 20, they're all dead. And that told us that, first of all, I'll say I was very surprised when we started doing these experiments at how beautifully reproducible these survival curves were. We did not expect that. I didn't expect that we could, that we would really find, to be honest with you, some specific cause of death in these animals. I thought that maybe, all kinds of things could go wrong, and so survival curves would be all over the place, but they were very reproducible. And it told us that perhaps something specific is happening around day 10 when these start crashing. And I'll show you what that is that's happening. But I'm going to show you first one other thing, which is that even if you look at the correlation of in individual animals, how much sleep they lost and how long they live, there's a really excellent correlation. So that animals that sleep the least, so plotted here, okay, animals that sleep the least are also the ones that die the earliest. Okay, so it's beautiful correlation in individual animals. Okay, so what happens here around day 10? So we figured out after, looking around, our idea was essentially to deprive flies of sleep and then dig around everywhere in the body and see if we could find any signs of cellular damage. And what we figured out is that there's this massive accumulation of reactive oxygen species, and I'll explain what that is, or ROS, specifically in the gut of sleep-deprived flies. So here, this row and this row are sleep-deprived flies, and then in columns you have various tissues, and you can see that, as compared to these other tissues that we've examined, including the brain, there's really a massive increase in reactive oxygen species. That does not mean that there's not increase in ROS in some specific neurons in the brain, and it's been shown by Gero Miesenbach's lab that there are actually some redox changes in the brain, but in very small number of neurons that are important for the regulation of sleep. But we don't see anything anywhere else like what we see in the gut. So this is massive increase. And we actually see it gradually coming on after about a week of sleep deprivation, and then it peaks at around day 10, and this is when animals are dying. Here is the quantification. So again, huge increase in the gut. But what is it that we're looking at? So the molecules that we're looking at are like this one here, like the superoxide. Reactive oxygen species, as the name suggests, they're extremely chemically reactive derivatives of molecular oxygen. So the most active forms excuse me, the free radical forms have a free electron in their valence orbital, or their outer orbital that engages in chemical reactions, and this is what makes them extremely unstable. So they're difficult to detect. You can't really detect them with anybody's stain. So what we did was to use chemical probes, such as this one, DHE, which get oxidized by specific kinds of reactive oxygen species, and then they become stably fluorescent. And that oxidized DHE is what you were seeing in the previous slide. So the reason why these molecules are dangerous is because they are unstable. They lack an electron, that paired electron in their outer orbital that would stabilize them. They steal those electrons from other molecules, from cellular macromolecules, destabilize them in the process, turn those guys into radicals, and that's how the damage can propagate. Oxidative stress can propagate. It's sort of like not unlike rust spreading to metal. So because raw steal electrons, they oxidize, like I said, other molecules, and that's what's called oxidative stress. We looked at a bunch of markers in the gut of DNA damage, specific kind of DNA damage, oxidative damage, at stress granules, which are accumulated aggregates of protein, untranslated RNA that occur in times of cellular stress. So essentially halt translation and wait for better times. Lysosomes, which degrade or recycle many of the cellular components, damaged cellular components, and then markers of that. They're all increased in these sleep deprived guts, meaning that there's massive oxidation. And again, we could not see a similar thing, for example, in the brain. So then the question was, as I told you, we used several methods of deprivation because we wanted to make sure that whatever we see is not specific to any one kind of suppressing sleep. So the next thing we did was to look at sleep mutants, and the various sleep mutants that we examined also behaved similarly. There was one exception that actually confirmed our rule, and we can talk about it later if anyone's interested. These were mutants in a dopamine transporter, and in that case, those guys were losing sleep, not dying prematurely, and this was actually shown by Rob Jackson's group 15 or 20 years ago when they cloned that mutant. So if you're mutant in a dopamine transporter, you sleep less, but you don't die prematurely. And what we found is that these are the only animals in which we could not detect ROS accumulation and oxidative stress, only sleep-deprived animals, and they're the only animals that don't die. And again, that's supporting our idea between ROS being causative for damage. But so it's for sleep mutants, we saw the same thing. And here we're looking at mechanical sleep deprivation, where we're just mildly shaking flies. And this it's very difficult. Anyone who's worked with flies and tried to deprive them by mechanical agitation will tell you that this is extremely difficult to do because sleep pressure is so strong, and so animals eventually fall asleep. So in most cases, this is what you see, where they're deprived on the first night, but then they just sleep afterwards. In some cases, in a minority of cases, we could see consistent sleep loss where flies were deprived on all days. And so here you see animals that are inconsistently sleep deprived, they lose much less sleep. And then we have some animals that are consistently deprived and lose a lot of sleep. And so the nice thing about this is that all animals are in the same setup. They're all being handled physically, but only some are deprived, some are not. But now you can compare their levels of ROS, and it can tell you whether any of the damage in the gut is actually coming from the shaking itself or from sleep loss. And I'm happy to say that only in cases where we consistently saw sleep loss, we could see ROS accumulation. That's meaning that it is really most likely lack of sleep and not physical stress that is causing these animals to accumulate ROS. Okay, we then asked whether this was a conserved phenomenon. Like I said, we believe that fly is an excellent model system, but of course, makes you feel better if we can actually see the same thing in mammals. And so we sleep deprived mice, and since my lab never worked with mice before this, we got help from Michael Greenberg's lab in the department, especially Liz Polina and Cindy Lin, who were just really amazing to collaborate with. So Here is the small intestine of a sleep-deprived mouse. Actually, the top is non-deprived mouse. This is after 1 day of deprivation, 2 days, and 5 days. So what you can see is that even after 2 days of deprivation, after just 2 days of deprivation, you see ROS increase. And I will say that we removed most of REM sleep and about half or 55% of non-REM sleep. So it wasn't a complete sleep deprivation, and we were only allowed to do it for 5 days. And when we looked at other tissues, we could not see the same thing, except in the large intestine. So here, this large intestine, and you do see some increase, and so that's quantified here. Small intestine, large intestine, there's a little bit of increase. Okay, and we also see oxidative damage and cell death. That occurs here after day 5. After 5 days, you see oxidative stress in the small. Okay, so what I showed you so far is that both in flies and mice, we see reactive oxygen species accumulation and oxidative stress in the gut. We also observe premature death in sleep-deprived flies. But the question was whether these things are causally connected. So if we were to block ROS accumulation in the gut, good survival of sleep-deprived flies. And so for this, we did a couple of things. First, we did a screen of antioxidant compounds where we tested 53 different compounds that we gave through food. We tested multiple concentrations, multiple different times of dosing, etc., to find good conditions. And we found for 11 compounds, we found that we could use them and actually suppress lethality in sleep deprived. So here again, we're looking at these two conditions in parallel where you're removing about 90% of sleep. The top two rows, and we're plotting the day of that for animals. So the top two rows are the parental controls. Then you have the sleep-deprived animals. Here you see that that's shifted to the left. These guys die earlier. With DMSO and ethanol, which are just solvents for our antioxidants, you don't really see anything. But then with these antioxidants here, such as melatonin or down to even vitamin C, I think this has less of an effect, but you still, with all of these, you do see a pretty decent shift to the right, pretty good rescue of survival. You're probably thinking about melatonin and wondering, if sleep is increased in these animals. I'll tell you that it's not. I'll show you in a second. But also, the melatonin in flies is not does not play a role in regulation of sleep or circadian rhythms. But what you may not have known is that melatonin is a potent antioxidant, which is actually found at many fold higher levels in the gut than it is in the brain, and it's not really clear what it's doing. Okay, so if you clear ROS, and which I'm showing you here, that some of these compounds, like melatonin, lipoic acid, and NAD, I'm showing you that they successfully clear ROS here. And so all the compounds that we're rescuing were able to clear ROS. The ones that did not rescue, we did not test them all. But the ones that we did not test them all for ROS, but all the ones that we did test, they all still had high ROS levels. So it seemed that if you could not clear ROS, you could not rescue survival. And here is sleep to show you that sleep levels remain low. Okay, so these are the non-deprived controls, and these are sleep-deprived animals. And then with antioxidants, you see that the levels of sleep still remain very low. Did not change the quality of sleep, the quantity of sleep. Okay, so we can say this is great. I mean, we see this really nice correlation that if we clear ROS from the gut, we can see animals surviving. But if we do it through food, then there's no guaranteeing that those antioxidants that you ingest don't act somewhere else in the body where we just can't see ROS. And so what we wanted to do was to specifically express antioxidant enzymes in the gut. And we can do that. We had to come up with two parallel, not come up with, but we had to use two parallel gene expression systems, one in the brain for sleep deprivation and the other in the gut for expressing antioxidant. So we screened through a bunch of Lexay lines, and Lexays are just like L4s, but they're this gene expression system, bipartite gene expression system that is similar to GAL4 but can be used independently. So essentially we use Lexays. We found Lexays that are expressed in the nervous system that you can activate, that label neurons that you can activate to suppress sleep. Okay, and so, for example, this Lexay line that we found, 5807 Lexay, when you activate the neurons labeled with it here in green, you actually suppress pretty much all the sleep. These flies, they're 100% is 100% suppressed, and they all die within 10 days. If we now, in that background, express antioxidant enzymes in the gut, we can ask what survival looks like, but not in the nervous system. And we can compare that to survival if we express antioxidant enzymes in the nervous system, but not in the gut, or I should say at lower levels in the gut. So some of the drivers that we use are expressed at very low levels in the gut, but some are not. So this driver, ELV-GAL4, is actually expressed at some level, at a very low level expression in the nervous system. In the gut, and high expression in the nervous system. And so here's what the experiments look like. So if you sleep-deprived flies with this Lexay line, like I said, they're all dead within 10 days. Here are deprived animals. Here are deprived animals that have a transgene, but that just have the transgene, the antioxidant enzyme, but they're actually sleep deprived. Sorry, they don't have the GAL4. They're sleep deprived, but they don't have a GAL4. These guys are sleep deprived, and they're expressing antioxidant enzymes in their gut. And you can see that survival is fully rescued. If we compare that to the results of overexpression in the nervous system, you get a very different picture. So if we're driving antioxidant expression throughout the nervous system, you don't get the rescue like you do here if you do it in the gut. You get a little tiny bit of rescue, few days of rescue here. You see even a shift here, which we think is real, it's reproducible, but that could mean that there's some contribution of the nervous system, but we're actually inclined to think that since there is a little bit of expression in the gut of this driver, ELV-GAL4, which I'm actually going to come to in the second part of my talk, then maybe some of this rescue is coming from the gut. But in any case, these two are very different. Sorry, I'm just going to check my email to make sure that I'm not logged out. It's really weird to not be able to get any feedback.
Everything is cool. Everything. feel free to ask for me.
Okay,
Really cool.
all right. So you guys
I'm
are
really enjoying it.
Okay. All right, cool. So, and this is just showing that sleep itself, again, is suppressed in these flies. So it's not like they're sleeping and that's why they get to live this long. No, they're still sleep deprived, but they just don't have ROS in their gut, and now they can survive sleep. And so what we're doing now, and I'm happy to talk about this afterwards if people want to know what it is that we found. I guess we'll have afterwards. But we're trying to understand what the cellular source of ROS molecules is. Because if we have a handle on this, we believe we'll be able to figure out what the signal is that comes to the gut, and that will explain why is it that these molecules accumulate when sleep is low. And then, so basically going upstream of ROS, and then we also want to go downstream, and we'll ask what is the impact of ROS accumulation on health and lifespan. In other words, what is it that kills you after ROS are accumulated in the gut? And I'm happy to say that we have already, we think we have an answer for where ROS are coming in the gut. It's from this enzymatic system. And again, I'll talk about that later if we have time, but I want to go very briefly into this other story. I think we have 10 or 15 more minutes, where I'll tell you the other way around. So now this was about sleep interacting with the gut, or the gut being sort of target of regulation by sleep. But here, I'll tell you that the gut also can influence sleep, and in this case, it's sleep quality or sleep depth. And this is a work of another really wonderful postdoc in my lab. It is Titos Ivankos. And she was helped by Ofer, Pavel, Casey, and Benjamin with some technical aspects. I'm sorry, I'm just going to ask for what. So, what Iris wanted to figure out is how is it that during sleep we are able to ignore much of our environment. How is it that we disconnect? In this case, how is it that we disconnect from the external world, from the world of senses? And this is something, of course, that occurs to all of us. Oh, not occur, that happens to all of us when we fall asleep. Right? We're in a different world, and we somehow are not plugged into the outside world to the extent that we are when we're awake. So to figure out, and I'll just say that we really know very little about this, about sensory eating during sleep in particular. So it's not really clear at what level these gates are set up, is in the nervous system or where this occurs, where you will actually prevent sensory information from flowing and arousing the brain to full awareness. So what Iris did was to build a system where she uses platforms. She uses these very large bass loudspeakers to produce vibrations, and then she has platforms on top of these speakers, and she has a program that allows her to control how much input goes into this. So you can control how much this platform shakes, a little or a lot. So you can provide more input here, so you get more vibrations. And so she showed that with lower amplitude stimulus, you wake up fewer flies than with higher amplitude stimulus. And so she used then to screen with RNAi for genes that could be controlling arousability in animals. So you use this driver ILA-Gal4, which is a nervous system-wide driver that I actually showed you just a moment ago, and you're using it to knock down individual genes. So we thought we were doing it in the nervous system, and you just did this with almost 3,500 genes. And then she looked at arousability. Okay, if you remove this gene, does it make fly more or less arousable than normal? And you can see that here with the low amplitude stimulus, most animals, actually I should explain, this is on the y-axis, we look at percent change in a woke-up fly. So the higher the number here, it means most animals woke up in response to that stimulus, and low number means few animals woke up. So here you see with low intensity stimulus, few animals wake up. And then here at the tail end, you see hyperarousable phenotypes. So these are flies here that wake up very easily, even in response to low amplitude stimulus. And conversely, if you use high intensity stimulus, you can look for hypoarousable phenotypes. So most animals wake up in response to that, but then you have some animals that can sleep through an earthquake, so to speak. And so I'm going to tell you a little bit about these two today, about these two genes. And they are CCHA1 and CCHA1 receptor. So peptide and its receptor, which if you knock them down with RNAi, both give you hyperarousability phenotype. So you see that many more animals now wake up here in response to stimulus than in the controls if you knock down CCHA1 or CCHA1 receptor. And each of these dots here is 8 flies, and you see the phenotype is very strong. We look at mutants of these two genes, we see the same. They're hyperarousable. So now very low amplitude stimulus, which wakes up 20 to 30% of controls, wakes up 90 or 80 to almost 100% of these animals. Okay, so where is CCHA1? Here, this is the staining. This is the brain. This is the ventral nerve cord, brain ventral nerve cord. This looks messy, but I'll tell you there's not that many cells actually that express it. So here are the cell bodies here. And then most of this stuff that you see here are actually dense core vesicles, or these vesicles that contain the peptides. Of the projections throughout the nervous system, but not that many neurons. And then if you target CCHA1 with RNAi, the signal is gone. So we thought, okay, great, RNAi works, signal, anybody works, whatever, we get a nice phenotype. But there was a problem. Actually, I'll jump right now and tell you that CCHA1 is actually expressed in the gut, and this is where our phenotype was coming from. If we were to remove CCHA1 just from the nervous system, you would not get a phenotype. Okay, this is where the phenotype actually came from, the gut. And this was problematic at first, like I said, because we could not figure out where the phenotype was coming from, because we saw it with ELA-4, but then if you were to target those neurons in the nervous system where you see CCH1, you would not get a phenotype. That's not where the phenotype was coming from. It was coming from the gut. And once Iris figured that out, she could, or once she figured out the CCH1 was expressed in the gut, she could specifically remove it from there and leave it intact in the nervous system, and then ask, what the phenotype looks like. And if you do that, if you remove it just from the gut, you actually see the arousability phenotype. So again, if you leave CCH1 in the nervous system, take it out from the gut, you reproduce the arousability phenotype. But there's no phenotype when it comes to sleep duration. So this specifically controls the depth of sleep. So what are these cells? Where is CCH1 expressed? CCH1 is made in enteroendocrine cells in the gut, shown here in orange. They are a cell population that's sparse in the gut. In case of CCH1, they are here in the posterior part of the gut. There's a specific population in this part here that expresses CCH1. These cells are, their apical side is facing the lumen of the gut. And these cells actually, they have characteristics of both epithelial cells and neurons. And so this is why at first we were fooled. Again, we used this ELA-4 driver, which people use as a neuronal specific driver. Okay, so we thought that our phenotype was coming from the nervous system. But because these cells in the gut are so similar to neurons, they're also expressing this ELA-4 driver. And actually, that's how we serendipitously stumbled upon this. Okay, so we were actually targeting a peptide in the gut just because we used this driver. We thought it was in the nervous system. So what could these cells be normally responding to? They're facing the lumen of the gut. And so we thought, maybe there's some food components that could be doing something to these cells. But first, we wanted to ask whether manipulating the activity of these cells, like I said, they're neuronal-like, so you can activate and silence them. So does silencing and activating these cells change arousability? And it does. If you silence EECs, and you can do it with the same tools that silence neurons, you make flies more arousable. So it's the same phenotype that you get if you deplete CCH1. So you can think of CCH1 as kind of a soothing peptide, so to speak, that puts you into deeper sleep. If you remove CCH1, you're much more arousable, and it's the same phenotype if you silence these cells that produce CCH1. You activate them, you have a soothing effect, you promote deeper sleep, you suppress arousability unless you remove CCH1. Okay, so if we activate the cells but don't have CCH1, then the effect is gone, telling us that CCH1 is actually mediating the effect of these environments. And fully responding to. How is their activity normally regulated? We asked if any of the food components, any specific nutrients, could be changing the activity of these cells. And we looked at this with this calcium reporter, transcriptional calcium reporter, which allows us to feed flies various different foods and then look post hoc at the response, what the calcium levels are. And so we did various sources of sugar, fat, protein. I'm going to show you just a few things here. If we use glucose as a source of sugar, coconut oil as a sort of fat, we don't really increase cell activity, these cells in the gut. But if we use peptone, which is protein, dietary protein, hydrolyzed dietary protein, then we see an increase in cell activity and we see increased. And it doesn't seem to be about individual amino acids. We thought at first it is, and I thought that maybe you use amino acids actually for press signaling molecules. And we thought that maybe, this is how food could kind of signal to the gut, but it's really not about that. It has tried various combinations of different amino acids, either individual amino acids or combinations of polar, non-polar, etc. And the only thing that seems to matter is total concentration, how proteinaceous food is. So if you're eating more proteinaceous food, these cells in the gut get activated and they produce more CCH1. And so the question is also whether that food, that peptone food, could actually can we see an effect on sleep depth there? If we're saying that these cells are activated in response to peptone to produce more CCH1, and CCH1 is the soothing peptide, then does this put you in deeper sleep? And it does. If you supplement regular food with peptone, and you use medium, here we're using medium intensity stimulus, okay, because that allows you to get both phenotypes that are up and down. We can compare, and in this case, you really see that sleep is, the depth of sleep is sorry, the flies seem to be in deeper sleep. There's fewer flies. And so here is, to summarize what I showed you so far. Okay, I'm almost done. That if when flies are eating regular food, you see there's still some CCHA1, but if regular food is supplemented with peptone, we see a lot more cell activation and a lot more CCHA1. And so this is, the idea is the dietary protein increases levels of CCHA1 in the gut, which signals to the receptor and suppresses arousal. And so the question is where the receptor is, and I don't have time to tell you how we found this. I'm just going to summarize and see if you guys have any questions. That in response to this protein-enriched food, again, CCHA1 levels are increased in the posterior part of the gut, of the midgut, and then CCHA1 actually signals to cells in the brain, dopaminergic neurons in the brain, and these dopaminergic neurons innervate the mushroom body. And so mushroom body is the structure in the brain that I'm sure many of you know, and which has been involved or implicated in the regulation of sleep duration. So the cells that make up the mushroom body, the intrinsic mushroom body cells or canyon cells, they carry information about that is relevant for the duration of sleep. And then the information from the mushroom body is picked up by only 34 output neurons. These mushroom body output neurons or M-bonds, which project to downstream centers. And so what we showed is that there's a sort of orthogonal pathway here. These dopaminergic neurons, which are known from many labs' work to modulate interaction between canyon cells and the mushroom body output neurons, actually also serve as a hub, so to speak, to receive visceral signals, in this case, information from the gut that's reporting on the physiological, nutritional status of the animal. And they seem to receive mechanosensory information, information about vibrations, which regulate their activity. And the activity of these neurons actually controls the level of arousability in flies, and there's no arousal threshold or sleep depth information in this part of the pathway. It seems to be contained here, and so that then the output neurons, the mushroom body output neurons that are leaving the mushroom body, they carry information both about sleep duration and sleep depth. So you have these two regulatory arms, the duration and the depth of sleep arms, converging on the mushroom body and feeding information to these output neurons. And so you can separately tune these two arms. And I will say that, not to confuse things more, but we do think that CCHA1 from the nervous system produced CCHA1 might actually be contributing to the regulation of sleep duration, but not CCHA1 from the gut. It has also been shown that CCHA1 is made in a particular set of clock neurons in the brain where it regulates the timing. Have these various separate aspects of regulation that are controlled by this peptide that it actually comes from different sources in the body and has different. And so I would just like to acknowledge some other people. So people here that are in purple or highlighted in purple have all helped with the sleep deprivation project. Alan and Alejandra are now have joined in the sleep deprivation studies and arousal threshold studies. I will say that Alan now is trying to do this thing that I just told you about with arousal threshold, to do those studies in mice. So this has not really been rigorously done in sorry, I was just going to stop sharing this. Okay. Let's see. Do you see me now, or I'm still sharing my screen, right?
You're still sharing, right? Maybe you can on top. Wait, in that.
Hold on. Close video. Is it gone? Okay. with us.
Yes.
So
No. Okay.
just because I was going to say that, we have this sort of intuitive or anecdotally, we have this feeling that, very food can impact your kind of how you sleep or how deeply you sleep, but this has not really been investigated rigorously. And so this is what we really want to do in a quantitative way. And so just to tell you how I'm thinking about this, because I mentioned to you that everybody says this, and I used to say it all the time, how sleep is dangerous and unproductive, this period, this weird period, etc. But I actually think that to me, it makes sense that if you had a rich, nutritious meal, you could actually afford to disconnect, sleep more deeply. You might be better off going somewhere and hiding and not having to run around and look for food. If you're fat, actually, you might be much better off. So that really makes sense, right? And we show that it's you have one protein-rich meal, it does improve your sleep quality a little bit. But if you're consistently on that kind of food, there's actually a more cumulative benefit. Your sleep really becomes even deeper and better. And so it's almost like your body knows if this is like a temporary trick. There's like just some blip in the environment, or if there's a constant source of food, and you don't really have to be stressed about it and go look for more nutrients. All right, I'm going to stop talking and see if there's any questions.
I was really enjoying. Really nice sum up of the whole your
I
whole
felt
presentation.
so weird, like not being able to. I usually feel like it's weird to see yourself on your screen, but it's even weirder to not see anything. Like,
Yeah, it's so
so
weird. Some
anyway,
people are not able to do it,
thanks
but
for
it's
making noise.
really cool. Okay, maybe we can go through some questions. We already have a few. So from Ignacio Rebolo, he said, "Thank you very much for this very interesting talk. I have two questions. So the first one is, what could be the brain mechanism responsible of gut antioxidant effect during sleep? Or in other words, why the gut needs sleep and the brain to regenerate?" And the second one, maybe you can answer this and I'll go to
Let
the
me
second.
answer that. So one thing I want to be careful to say, and you'll see that I didn't say that this is that this is the function of sleep. So I can't tell you that for sure. What I can say is that I think we figured out what kills animals when they're sleep deprived. I can't tell you for sure that that's normal function of sleep. It's possible that we're creating some uniquely adverse conditions that are killing the animals. So we don't know yet if there's some rhythm of oxidation, for example, that needs to happen in the gut. We have not found evidence for that yet.
Actually, yeah, no one has checked, like, for example, if animals that sleep more versus animals that sleep less, like these levels of ROS with time.
We tried to look at just a baseline to see if there's any oscillations, and let's just say we didn't see anything obvious at least.
Okay. Yeah.
I can't
Okay.
say that this is really, the daily function of sleep, but it is that when you prevent animals from sleeping, this is what happens, and we still want to figure out why. What is the signal? And we don't know that yet. We don't know yet what triggers it.
So yeah, his second question is, if you could study these questions in humans, which experiment would you do?
So what I would really like to do is to find some markers in poop, in feces, that we could use to measure like inflammation, or we're actually trying to, if anybody has a good idea of what we could look at, that's what we're trying to do, essentially find some, we're looking for things, maybe if there's a good guess, but we'll also try to do some unbiased or some, kind of, yeah, unbiased ways of just like analyzing, are there any molecules there that we could use to then get a bunch of, undergrads like around exam time, cramming, and then see if we see anything because I think it's really, there's like so many of us have this issue where we don't sleep enough. I mean, I studied this stuff and I still don't sleep enough because it's just so tempting to stay up at night. And so there are products that are, that are made like to help you sleep better and stuff. But it's not like for many of us, it's not like we couldn't sleep better, like we just don't. Sometimes it's work, sometimes it's whatever, parent care, child care, whatever it is. Okay. But for all of those people, and even people who think, I don't need more sleep, I'm fine, that, your body still may need more sleep. And there's no treatment that could actually help you offset some of those negative effects. It's nothing like that. So we want to, first of all, figure out, can we see some reporter of, things that are happening in the gut in humans, and then actually like create some therapy.
That's interesting. But I'm sorry.
Sorry. No, sorry, go ahead.
You would have to correlate kind of people sleeping with their, it would be like more or less subjective, right? It would never be like something super, it would have to be more or less a correlation.
There's some people that have that study actually human sleep here in the division of sleep medicine. So we have talked about, like they actually
I see, really using the, okay, because
we thought even about, really just doing like undergrads during, just from getting some volunteers, and when they're like not sleeping that much, during the night for a few days, see what happens and stuff like that. But this reminds me, actually, I want to make another point, which I think is really important. So many people say, I'm fine with like 5 hours of sleep, 6 hours of sleep, whatever. So I will say, what you mean by saying I'm fine is that you don't feel so crappy, like mentally you're okay. But really, you have no idea what's happening to you in terms of like this physiological effect. I think you may have some people more sensitive, they can tell, some people can't tell. And what's important is that because of this method that we use to activate neurons to suppress sleep, based on these lines that Bill Joyner's lab found, that was really crucial, because there you can suppress sleep without triggering the homeostatic sleep response. That was really clear, that crucial that they found those neurons that can be activated to suppress sleep, but without the animals kind of feeling it mentally, so to speak. This is a little bit interpretation, but they don't show signs of really wanting to go back to sleep. Okay, still you see this ROS accumulation in the gut. You also see it in animals like mechanically deprived animals, which clearly have a normal homeostatic response. You also see the same thing. But again, even in animals which seem to be fine from the perspective that they're not trying to go to sleep as soon as they can, they are also paying that price.
Really interesting. Okay, we have some more questions. So now from you to Yoshinari, he said, thank you for a wonderful talk. I would like to ask about the action of gut-derived CCH1. Does CCHA1, from the gut directly activates these sperm gamma-3 dopaminergic neurons after passing the blood-brain barrier? Does any brain CCHA1 neuron project to the dopaminergic neurons?
So we didn't really notice any projections, but there are CCH1 neurons in the brain, and like I said, we think that those are actually contributing to the duration of sleep, but I'm not sure how that goes. I'm not sure about the projection. It doesn't seem like they're contributing to this thing that we're studying in terms of sleep depth. That really seems to be from the gut.
Cool. Okay, then we have a question from Krishna Melnatur. Thanks for a lovely talking story. I had a question about cognitive defects following sleep loss. Does blocking ROS accumulation rescue cognitive defects following sleep loss? Cool.
Hi, Krishna. I know him.
Okay.
Yeah, that's great question. We're actually wondering that, and we're doing some experiments now to address it. So I don't know. The question is whether these animals that are rescued, whether they're, also cognitively better, they're living, but they're still stupid, or like not at their top cognitive performance. I don't know. We're doing these experiments in mice now, where we gave all the mice we're approved now for longer deprivation for 15 days for mice. So we gave them to this behavioral core where they're testing, and we see some clear defects in sleep deprived mice. So we're going to try in mice actually to target that. We haven't done the thing with flies yet, but eventually we should test some things as well. I'm just looking for really robust assays that we could test, because behavior is a little bit finicky. But I agree, that's a really good thing to answer.
Cool. Okay, we have some more questions. Okay, do you have a theory for why SD in the human mutants does not lead to gut ROS accumulation and then lifespan?
Yeah, so we have, we have some thoughts. I don't know the answer yet. So dopamine itself is also an antioxidant. So we thought that maybe in a dopamine transporter mutant, what happens is that you can't clear dopamine effectively that's been released. So there's more extracellular dopamine sloshing around. That's one of the things. There could be other things. So we thought maybe dopamine, because it is an antioxidant itself, it could be directly neutralizing, like we did with melatonin can do that, or some vitamins. So we tried supplementing with L-Dopa, which is a precursor that can be converted to dopamine by DDC, and we could not see any rescue. So we don't know if that's because maybe it's not effectively converted to dopamine, or it could be that dopamine is doing something else. So for example, the converse is the dopamine itself can get oxidized, and then this is one of the ways that some of the neurons get destroyed. So actually the dopamine can get oxidized, and it becomes kind of a free radical. But what happens is that if you have chronically slightly elevated dopamine levels, such as you would expect from a dopamine transporter mutants, then you have sort of slightly elevated oxidation levels, baseline oxidation, and then actually that turns on some protective pathways, some antioxidant protective pathways. So there's this idea that a little bit of stress is good. And what does that mean, right? So a lot of stress is bad, but a little bit of stress is good. And it could be that if you have this condition where you slightly just upregulating some protective pathways, you may be better off in the long term. But I don't know yet what the answer is. But yeah, that's another thing that we would like to know very much what the answer is.
Cool. Yeah, that would be super cool to compare these two models, or I don't know, even molecularly to see what could be different.
Yeah, definitely.
Okay, so now we have another question from Terufumi Fujiwara. He said, "Thank you very much for the great talk. Do you have any idea what is the functional difference between the mushroom body and central complex in sleep regulation?"
So, well, I mean, they are different in terms of where they are, I guess, in the executive order. So I would say that you have to first process a lot of information probably through the mushroom body, and then you go to the central complex to kind of inform sleep. But before that, you sort of along the way, you have to gather information that's going to be relevant for informing, how long sleep is going to be, how deep it's going to be, all of this sensory stuff. And then at the central complex, you're probably getting other inputs from other places that eventually drive behavior.
Cool. Okay, now from G. Batista, great talk. I was wondering if you see any behavioral deficits after sleep deprivation and whether they are reversed by antioxidant treatment.
So we already
it's kind of
talked about
similar.
We do. I mean, we do see, but we don't know yet if it's reversed.
Exactly. Okay, another question from Ben. Hi, Dragana. Temperature variations have also effects on sleep, as you showed, right? I'm wondering, why did you keep thermogenetic activation, which adds a change in temperature, on top of a neuronal activation instead of optogenetic activation? Thanks. Okay.
Well, because light also affects. If we were using light, if the lights were on the whole time, then that would not be that great, because then that has effects on circadian rhythms. That's why.
Yeah.
But we do think about this. We were worried about it. Like, okay, we don't want something that's specific to temperature. So that's why we had the mutants and mechanical deprivation in mice, and in mice you don't. It's also there's no temperature change, so we don't think it's because of that. I'd like to avoid it, but we haven't found anything that's better.
Yes. Cool. And now a final question. Why do you not get rebound sleep with the tool golfer drivers you use to induce ST?
We don't know that yet. That's another great question. Like I said, a lot of people are interested in understanding how the homeostatic response is triggered. And again, this is Bill Joyner's lab found these lines. But what is really interesting again is that you can separate those things. So a drive to sleep, a drive to eat, it's a trick that your nervous system creates, right? So I could suppress your appetite. You could feel no hunger, right? You're still going to die because your body needs nutrients. In this case, you can suppress sleep. They don't feel it. They don't feel bad, but they still need the sleep. So this was really critical for us to but I don't know why. I don't know why those particular animals don't get. So I think Bill's lab is probably very interested in that. I'd like to know. I'd like to know myself, but I don't.
It's really interesting. I actually had one question of myself that I was wondering if anyone would ask, but no. It's actually about the gut microbiome, right?
Okay.
If you actually think that the gut microbiome somehow has the capacity to regulate these oxidation levels in the gut
Yeah.
and help promoting sleep, maybe.
Yeah. So actually, okay. So the enzyme that we found that actually sorts of the gut of ROS in the gut is this NADPH dependent oxidase. We're also testing it in mice now, and we have indication it could be the same thing actually in the mouse. And so that particular enzyme can be activated by lactate, which can come from bacteria. And so we did think that maybe there is something like that. We've tried, we haven't profiled the microbiota yet. We're doing that now with flies. But we've tried giving some antibiotics to flies and mice, and we couldn't really see any effect, any change. But we haven't done it super extensively. Now, lactate, like I said, in this case, the enzyme that we're looking at, it's called NOx. It's actually enriched in the gut, and it is activated by lactate, which would come from bacteria. But lactate also is a, metabolic.
Yeah, yeah.
So it could be coming from other places in the body. We're actually trying to chase down the source of that lactate now. So we don't have any evidence for this, but of course, people know that there are changes in bacteria that happen during sleep deprivation, but we don't know. It's not clear whether that comes before or after us. It could be just that after you have this extensive oxidation, your gut is obviously messed up. I'm sure that the profile of microorganisms in your gut is going to change afterwards, but I don't know if it actually precedes any of it.
Of course, it's always the egg and the chicken. Yes, really interesting. Thank you so much
Thank you
for a
so
very
much.
interesting talk and discussion. It was really,
Thank
really
you.
fun.
So I'm
Thank
going
you,
to the Zoom
everyone.
link now. Now I go
Yes,
to the Zoom.
now we'll go to the Zoom link, and I would like to thank everyone for the questions too and your curiosity. And next week we will hear from Dr. Douglas A. Bayliss about the retrotrapezoid nucleus, an integrative and interoceptive hub in neural control of breathing. And with this, I'd like to thank you and see you next week.
Thank you
See
so
you
much. It was
soon,
great.
Dragana. Thank you.
Thank you, guys. Bye. Thank you so much for inviting me. Bye.
Bye.